extranuclear inheritance, and the examples. History about organelle heredity. E.B. Wilson, Carl Correns, Boris Efresi, Ruth Sager, F. Meinhardt, and H. Schriss. Characteristics of organelle heredity. Organelles heredity, which is mitochondria and chloroplast. Endosymbiotic theory. The origin of mitochondria and chloroplast from free-living prokaryotes. Mitochondrial DNA, mtDNA. The structure of mtDNA. Mitochondrial DNA replication. The detailed processing steps of mtDNA replication. There are 5 steps of it. Diseases caused by mtDNA mutations occur in 1 per 1,000 people. There are 5 syndromes of mtDNA mutations. Causes of mtDNA mutations. Those diseases are NARP syndrome and LHON syndrome. Furthermore, Melas syndrome and Lee syndrome. MERRF syndrome is a multi-system disorder known as myoclonic epilepsy with ragged red fibers. Mutations on MERRF cells and the consequences. The diagnosis and treatment for MERRF syndrome.
Let's know about cpDNA.
The genome organization of chloroplast DNA. The gene content of chloroplast DNA. Learn about replication and inheritance of cpDNA. Next, infectious inheritance. Introduction about infectious inheritance. The characteristics of infectious heredity, non-nuclear inheritance. It is a non-Mendelian inheritance and is in uniparental transmission. Examples of infectious heredity. Kappa particles in paramecium, rod-shaped bacteria that live symbiotically within the host's cytoplasm. Altenberg, S. Evidence, 1948. 38 degrees Celsius kills kappa, but not paramecium. Inheritance of kappa particles in paramecium. Aurelia. Role of nuclear gene. Dominant gene express the killer trait. Recessive gene sensitive to paramecin. Phenotypic expression. K gene in normal cytoplasm is potential killer. Sensitive strains cannot maintain kappa particles and have no immunity to to paramycin. Nuclear cytoplasmic interaction, exchange of gene and cytoplasm. This is the exchange of gene and cytoplasm in the nuclear cytoplasm interaction using killer and sensitive strains as an example. Wolbachia bacteria in insects. It is first discovered in 1924 in the common house mosquito by Marshall Hertig and Simeon Burt Wolbach. Advantages are vector control, disease suppression, and sterile insect technique, SIT. Let's go to maternal effect. What is maternal effect? It is a situation where the phenotype of offspring is not determined by own genotype. It is result from asymmetric distribution. Sperm contributes only chromosomal DNA. Zygotic development is directly affected. Let us know more about maternal effect and the leading statement of it, which is evolutionary changes in offspring traits. Characteristics of maternal effect. Maternal effect mechanism. Gene products transferred to oocyte. These products influence early development. Challenges and limitation. Complexity in genetic studies and dependency on maternal health. First benefit of maternal effect: enhanced development success. Second benefit is facilitation of evolution. Immediate impact. This allows for immediate environmental adaptation. Next, axis formation. Guide the spatial organization of cells and tissues. Third, provision of nutrients and resources. Support the growth until the embryo obtain own resources. Examples of maternal effect. This is simple explanation and demonstration of maternal effect using water snail as an example. The history studies of maternal effect. A.E. Boycott studied this phenomenon, and Alfred Sturtevant explained the findings. The references. Big thanks from Group DNA Dynamos.